So today I'm going to explain my understanding of the central dogma, which is gene expression by transcription and translation of the genetic code. So this comes from Unit 3, Outcome 1, and I'm using slides from Ed Rollo and Khan Academy because I think they're a really great resource and it's going to help me visualize my understanding. So we're going to be looking at the structure of DNA, the three forms of RNA, the similarities and differences in the subunits of those two genetic codes, and the gene expression in transcription, processing, and translation. Within the dot points that I'm looking at, I have left out condensation polymerization because that's actually discussed a couple of dot points up. So we would have gone through that before this sequence, and also the redundancy of the triplicate code, which I don't quite have time to go into. So let's begin. Starting off, this is a nucleotide, which we're looking at here. And if we drew a line through here and we looked at this side, we'd call it a nucleoside. So these couple of rings here create a nitrogenous base. We've got our sugar, which is this numbered structure through here, this pentagon with the fifth carbon as a tail. And we've got our phosphate group, which is this one here. So phosphate being the orange and the oxygen being the red. And we've got some hydrogens attached, which are the little white dots in this structure. So what we need to understand from this is our numbering of carbons, which is numbered here for us. So we always start at the carbon that's attached to that nitrogenous base. We then go clockwise and we number all of our carbons, remembering that we won't number this oxygen here because it's an oxygen and not a carbon. So what we see from this is 1, 2, 3, 4, and 5 carbons. Our nitrogenous base is always connecting to the first carbon. Our phosphate group is always connecting to that fifth carbon. And that's why we've got the number 5' up the top here. The other carbon that gets a phosphate group attached to it is this 3', which is why we've got 3' down here. So this oxygen or hydroxyl group down here is actually going to create this on the next whole nucleotide that will join the sequence. So this side is looking at base pairings. So within DNA and RNA, we have 4 nitrogenous bases. They are different in each DNA and RNA. So DNA has an A, a T, a G, and a C. And in RNA, we still have G and C. However, we have A paired with U. So we've got adenine, guanine, cytosine, thymine, and uracil. And we use the first letters of each of those respectively. The other thing we need to know is that these here are hydrogen bonds. And you can see that one pair down the bottom here only has 2 hydrogen bonds holding it together, and the pair up the top has 3 hydrogen bonds holding it together. So 3 hydrogen bonds holding it together are guanine and cytosine. So this is a GC pair. Down the bottom, we have adenine and thymine pairing, with thymine on this side and adenine on this side. So the other thing we need to know about DNA structure is that the single rings are called pyrimidines and the double rings are called purines. Uracil in RNA, so if we swapped these out and had RNA, we'd still have the same G and C pairs. These would look nearly exactly the same. And we've got A and T, but remembering that RNA doesn't have a thymine, it has a uracil instead. So here we'd still have an A as our purine, but our pyrimidine pair would be uracil. So it would have a slightly different structure here. We're going to use this slide to look at the differences and similarities between DNA and RNA. So the first and most obvious is that RNA is single-stranded while DNA is double-stranded. Now, we did talk about pairing in the last slide, and that's because RNA is used as a messenger, as a translator from the DNA, from the genetic code, and that gets processed. So that's when it starts to pair. We're going into that a little bit later. So that's the first main difference. The second main difference is that on this second carbon down here, and if we look at the same second carbon down here, we've got a hydroxyl group attached. So that's where we get deoxyribonucleic acid and ribonucleic acid. So the deoxy is within the DNA because it doesn't have that hydroxyl group attached to that second carbon. The other thing is thymine and uracil that we discussed in the last slide, and that's this one down the bottom here and this one down the bottom here. So DNA is going to have a T or a thymine, while RNA has this uracil, and we can actually see that there's a difference in structure in this part of the nitrogenous base. So they're the three main differences in DNA and RNA. So here are the three different types of RNA. So RNA always exists as that sugar phosphate backbone and the nitrogenous base. However, they have different roles within transcription, processing, and translation. So I've got messenger RNA, which is this long strand, which feeds into a ribosome, and the ribosome has rRNA or ribosomal RNA. And then we also have transfer RNA, which are these little boomerangs here. The reason they form different structures is because they still want to have that hydrogen bond, so they actually kind of fold up on themselves. So mRNA, rRNA, and tRNA. So as a quick overview, we're going to look at the processing of transcription, processing, and translation to read the genetic code from DNA. So we have the process of transcription and the processing of the product of transcription in the nucleus. And then we have translation occurring out in the endoplasmic reticulum, which is where all the ribosomes are. So ribosome hosting rRNA, ribosomal RNA, and transfer RNA is in the cytosol as well. And we have our messenger RNA created in the nucleus and then pushed out of the nucleus into the cytosol to be translated by the ribosome. So this is the process of transcription. So transcription begins when an RNA polymerase finds a tartar box, which is a repetition of thymine and adenine base pairs within the DNA. So what happens is that RNA polymerase attaches to that at the tartar box and pulls it open. We have a coding strand along the top here and a template strand along the bottom. What happens is this RNA polymerase pulls free nucleotide base pairs that are floating around in the nucleus, and it pairs them to create this strand here. This strand here is called pre-mRNA because it hasn't been processed yet. So it's not the actual strip of RNA that's going to be translated into a polypeptide chain. So what's happening is this RNA polymerase is moving in a right-hand direction and the DNA is being wound up again behind it. So this is the processing of that pre-mRNA. So we have these hollow spaces in here and these coloured spaces here. The coloured spaces are exons because they are going to be expressed, and the non-coloured sections are introns, and they're going to be spliced out to create a shorter sequence. So you can see in that bottom line there that we've had those introns spliced out, and you can see that we've actually added a poly A tail and a guanosine cap. So that guanosine cap is going to go into the ribosome first and make sure that while we go into the process of translation, that everything's going to be in the right order. Because it does code for proteins, we need to make sure that we're reading the right way. It's like reading a sentence backwards. It doesn't make the same sentence. So this is a diagram of the process of translation. So this is where we express our genetic code. So from capping the 5' end, we've gone in, we've fed in this way, and we've started with this triplicate here. So this triplicate is called a codon. A codon exists in the mRNA, and it's then paired with an anticodon, which is this here. So they're exact opposites. This is when that hydrogen pairing or hydrogen bonding between the two nitrogenous bases occurs within RNA. This is when it becomes double-stranded for an instant. So what happens is this feeds into the ribosomal RNA. Ribosomal is our only non-coding type of RNA. So what happens is this anticodon is attached to an amino acid. In this case, UAC codes for an amino acid met. And then the next codon, which is the next set of 3, is UGG. You can see that this tRNA is bringing an ACC into that space. So this is where we have that transfer RNA. This is transferring this amino acid onto the next bit of the chain. So this is going to create a chain here. So this is just another representation of that translation process in gene expression. So we're going the other way. So this is actually the 5' end and this is the 3' end. You can see that this is the tRNA that I was talking about before, that's sometimes represented like this, and it's pairing these codons and anticodons respectively, pushing it through to the next section in the ribosome and creating this polypeptide chain. So the polypeptide chain is also joined by condensation polymerization. That process is covered in a dot point up, which is why I haven't gone into it in this sequence here for my understanding. From here, the next step is that this polypeptide chain creates proteins, which are different shapes, and those kinds of things affect a whole lot of stuff in biology. So that's the next section that we would go on to.